Good morning, everyone. I am Marcela Mendes, professor of the School of Agronomy at the Federal University of Goiás. And it's an honor to welcome you all to the third edition of GBI Day. This event is organized by the students of the Precision Breeding Lab, LAMP, from UFG, coordinated by Professor Rafael Tassinari. We are backed by the support of the graduate program in genetics and plant breeding at UFG. Our partner study groups, Eugene from UFG, Jane from UFLA, Gevenk from Exalc USP, and Jane Melhor from UFB. And they start up the program. A warm thank you all. Our main goal is to bring together experts from all over the world to discuss the latest advancements in genotype by environment interaction. We believe that G-by-E interaction lies at the very heart of the most pressing challenges in modern plant breeding. Understanding these interactions is essential to developing productive, resilient, and stable plants, especially in face of climate change and the growing global demand for food security and yield quality. This year, our focus is centered on predictive models for plant breeding. We remain deeply committed to hosting an open, inclusive, and collaborative event that brings high-level scientific discussion to everyone seeking knowledge. We are thrilled to share that for this third edition, we have reached over 800 subscriptions. Welcome to everyone joining us from every corner of the globe. Before we begin, I have two quick housekeeping announcements. We will be using a dedicated link for each lecture. At the end of each section, please migrate to the next link, which will be posted directly in the YouTube live chat. Feel free to type your questions in the chat at any time. We'll pass them along to our speakers at the end of the presentation. Our first lecture will begin in just a few minutes. So please stay tuned while we get ready. I wish you all an insightful, engaging, and fruitful GBI Day. Okay, good morning again, everyone. We will now begin the lecture presented by Professor Fred Eeuwijk. I hope I pronounced it correctly, yes. Professor. Dr. Fred is Professor of Applied Statistics at Wageningen University and Research, specializing in statistical genetics and the modeling of genotype by environment interaction in plants. His work stands out for integrating quantitative genetics, statistics, and crop physiology to predict complex plant phenotypes. Recently, his research has focused on longitudinal modeling to capture dynamic, time-dependent trait expression using high-throughput phenotyping data. Furthermore, he combines deep neural networks with mixed models to enhance predictive accuracy in multi-environmental trials. Professor Fred, it's a true privilege to have you with us today. On behalf of the entire organization and our global audience, thank you so much for sharing your time and expertise with us. The floor is yours.
Thanks a lot for the introduction, and thanks a lot for having me. I mean, it's a pleasure, let's say, to offer a presentation for this audience. So indeed, as you said in the introduction, I will be talking about G by E, especially about what can high throughput phenotyping data add to, let's say, understanding and modeling of G by E, and also what are the opportunities here for AI. Okay, let's see whether I can move forward. Yeah. Okay, before I start, I also just wanted to, let's say, place the attention on a meeting that we will have in November in Australia. This slide will come back at the end, but I just mentioned it here at the beginning. Okay, so what is our story about? Well, if you look at introductory texts about genotype by environment interaction, you often find the kind of cartoons that you see over here. There is something with DNA, there is something with the environment, and by, well, something magical, there are phenotypes. And actually, although, let's say, I think we are working already for a century on modeling phenotypes in terms of statistical models, I think so far it has been really hard, and I think only recently, I think we're starting to get a finger on what's going on. Some recent cartoons look more like the one you see at the bottom, and there are more arrows, but I also have to add that most of these cartoons, don't tell you a lot about what actually is going on or what you could do in terms of modeling. So let's look at the questions that I will be addressing this talk. So I will give some overview about methods, especially, of course, it's biased towards the methods that we're using in Wageningen. I will say something about how I think we can use longitudinal information. I know that there are a number of options, but I think more or less I cover the options that are there. I know there are some competing options, but they are not very far off from what we try to do. And then I also will say something about the possible role of artificial intelligence in all of this. And let me state beforehand already that I don't think that the use of artificial intelligence models is very different of what we were used to do in terms of statistical or mathematical modeling. Okay, let me see. Yes. Okay, so let's first introduce some so-called reference models. I mean, the basic model that we often use for getting a first impression of the genotype by environment interaction is, let's say, fitting two-way analysis of variance on mixed models of the type that you see over here. We have the phenotype for genotype I, environment J. I will use I for genotype and J for environment everywhere in the talk. So there is some intercept, a genetic main effect, environmental main effect, a G by E term, and some residual. And of course, using these kinds of models is, well, okay to get a first impression of what's going on, but doesn't bring you very far in terms of prediction. So if we want to understand better and predict to future environments, we need to be able to find the environmental drivers. And what I call here XJ with superscript E is mainly an environmental variable, environmental driver that is defined on the trials that we're looking at. And this theta I is a parameter related to the genotype, and it can be anything, but most often it's a kind of sensitivity to environmental drivers, let's say, that are captured by these X's, these XJs. And there is some residual and, well, a genetic residual, and also some, let's say, trial residual. Okay, now, what within our group we have been looking at a lot, let's say, over the last years, is on the use of differential equation models for modeling, especially the dynamic traits that nowadays we can model using high throughput phenotyping devices, also satellites. This looks a bit different because instead of modeling yields or whatever kind of trait itself, we model the change in yield with respect to time or biomass or plant height, whatever. And this R is just a general expression for the function, the differential equation model that we're going to use. Well, these differential equation models are slightly different from, let's say, most statistical models in that, of course, they have explicitly time in them. And this Y vector, this is a vector of phenotypic traits that change over time that we may need, let's say, to model the change in any trait we're interested in, typically biomass or yield. And here, once again, we have only the environmental covariates that are necessary, let's say the environmental drivers of change. And then here at the end, we have the same rate parameters that are genotype specific. And actually, what I think our task is to figure out what kind of function R is describing the development, and then what kind of environmental drivers do I need, let's say, to model the change in biomass or yield, and then what are the differences in the rates for the individual genotypes. And finally, because we're doing genetics, we would like to be those rate coefficients, those sensitivities to the environment. We would like these to be functions, simple functions of DNA information, DNA variation. And so typically we model the rate coefficients or the genotype specific parameters in terms of DNA variation, marker variation, etc. And that's expressed by this expression over here. And of course, a very important question is, well, which environmental characterizations really do drive my phenotypes? And of course, well, typically we have soil information, weather information, management. These can be quantitative and qualitative. And nowadays, all of this also is accessible to us via satellites, drones, sensors in the field, etc., and also at probably high time resolution. Okay, now, if we look a little bit more at dynamic and static models, I mean, if the change in a particular trait Y, which could be biomass, if the change in biomass with respect to a change in a certain environmental driver, and X could, for instance, be the amount of water available, it could be, well, let's say, accumulated temperature, growing degree days, whatever, then if this rate over here for a particular genotype is constant, it actually means that for the state variable biomass, we could use a simple regression model like we see it over here. And so this dynamic and this static information, or this static and dynamic formulation, are actually equivalent. But in statistics and genetics, so far we mainly looked at the static representations. And nevertheless, I think many of us are aware that when we use environmental covariates in statistical models or mathematical models, typically those models are already integrations over a certain period. For example, it's the amount of radiation that was captured by the canopy or by the plants between, well, flowering and harvest, between let's say initial phase sowing and flowering, whatever. And so what we need to figure out is which periods are critical and which environmental variables then do we need to integrate for us to be able to build either those type of regression models or those type of dynamic models. Now, maybe it's good to realize that our standard two-way analysis variance model, if we accept that it could be expressed as, for example, the sensitivity with respect to a certain stress where, for example, well, let's keep it simpler. We have certain resource. So this might be the amount of radiation available during the growing period, and this is simply the radiation use efficiency. Well, if we use this model as the underlying biological model, very simple model, and we would apply a two-way analysis of variance to data coming from this model, we would end up with a genetic main effect that is mainly the sensitivity of the genotype with respect to the average radiation across all environments included in your analysis. And the environmental main effect would mainly be the average sensitivity across all genotypes times the environmental covariate value for a particular environment. And the G by E would be defined by the eccentricity of the sensitivity of the genotype times the eccentricity of the environmental covariates. So you see that this regression ID, this rate ID, actually is underlying very much of what we do already in static ANOVA and linear mixed models. If you think about the typical expression for interaction, means, well, interaction often is interpreted as the difference between genotype. Do I do this right? Okay, this is in the so this is the difference for genotype I in environment J and J accent. This is for another genotype. Well, if the difference between those two environments for genotype I is different from the same difference for another genotype, then actually we do have interaction. And once again, it's easy to see that actually this boils down to the genotypes being differentially sensitive to environmental drivers. So any non-parallelism of, let's say, the reaction to the environment indicates actually G by E. And well, here you see a number of cartoons that come from this paper here on the left. And all of the cartoons here at the bottom show different examples of genotype by environment interaction. Those lines, well, we refer to them like reaction norms, which mainly means sorry, the translation of the environment, of an environmental gradient into a phenotype. And you can see that as soon as these reaction norms start to diverge, converge, intersect, or let's say more complicated patterns, we know that G by E is going on. Now, at this moment, most popular models for the analysis of genotype environment interaction are actually related to linear mixed models. And if you look at how we could do that, well, standard genomic prediction basically is regressing a genetic main effect over the full set of markers that is available. So these X's subscribed by G or superscripted by G, they refer to the marker information. These are the allele substitutions. And when we add those over all possible markers, and we end up with the prediction for the genotype. And if we make a simple assumption, well, typically we think that, or we don't think, it's convenient for us to think that the slopes, the allele substitution effects come from a normal distribution. Well, this over here is standard GBLUP, or not, well, this is a rich regression, but let's say this is a regression form of what we often call GBLUP. Well, for the environments, we could do actually exactly the same, but very often we don't do so. And we could also try to describe the environmental main effect by a regression on all kinds of environmental covariates. And then once again, we could pretend that these slopes all come from a normal distribution. And for the G by E, most of the models are nothing else than taking the products of markers with environmental covariates, estimating a proportionality constant. This is just a constant to either shrink or blow up this product, and average this term is averaged across all markers and environmental covariates. And once again, to keep life simple, we make an assumption that the slopes, these proportionality constants, they come from a normal distribution. Well, effectively, this mainly means that we replace the original analysis of variance model by regressions on markers, environmental covariates, and their products, and we make three additional assumptions on that the slopes come from normal distributions. Now, increasingly more popular is the following expression, formulation of the same model, and once again, it's not necessary that you understand all the mathematical detail here. This is exactly the same model. We still regress the genotypic effect on markers, the environmental effect on environmental covariates, and the G by E on products of markers and covariates, environmental covariates. The only difference is here that the modeling takes place via the relationships between the genotypes. So the idea is genotypes that are more alike with respect to their marker profiles will also be more alike in their phenotypes. Exactly the same happens for the environments that are more alike with respect to the environmental drivers, well, will also have phenotypes that look more alike. And for the G by E, it's simply taking the similarities between the genotypes based on marker profiles times the similarities based on environmental drivers, and those products, well, determine the contribution that the G by E will give to the total phenotypic variation. So this is exactly the same model as on the former, but this model is more flexible in the sense that it can be connected to other ways of creating models. For example, Gaussian process model, but also deep learning can be formulated in exactly the same way. Okay, now to make it less complicated, let's go back to a somewhat simpler model in which we start off with analyzing data in a two-way analysis of variance mode. And then the first thing we do is we replace the G by E by regressions on environmental drivers. So those XEs over here could be humidity, temperature, could be anything that defines differences between environments. And the betas, they are genotype specific, which means that the genotypic sensitivities, well, they create the G by E with respect to those environmental drivers. If we go one step further, we could ask ourselves, well, it's nice to have genotypic sensitivities with respect to environmental drivers, but can I find a genetic base for those sensitivities? Well, we can. If we do that, actually, once again, we regress those sensitivities over here in this vectorial regression model. We regress it on marker information, and so we predict the sensitivity from a marker profile. If we do that, then actually the G by E will look like we see well, down here. It's product of marker information times environmental driver, and then we estimate a proportionality constant. We can do exactly the same for the genotypic main effect once again, and I explained it already, but in this case, if we don't include all the markers, but we would, let's say, try to identify a limited number of markers that explain or are important for the genotypic differences, well, in that case, beta is a QTL allele substitution effect, and the betas over here, they're actually referring to QTL by E. I already indicated that in principle, we could also try to understand the trial differences by regressions on environmental covariates, but so far, very few people try to do that. An example of, let's say, a paper of a few years ago in which we, let's say, applied this technology is given over here. This is related to the EU DROPS project. It's on identifying genetic basis of drought stress in maize. And here we have an example of a QTL in which the allele substitution effect decreased with the soil water potential in the particular trial, and also in a nonlinear fashion. So, I mean, these relatively simple regression type of models can be already quite flexible also in modeling nonlinear environmental dependencies for genetic effects. Okay, now I have to go to the next slide. Okay, so in the same project, we went one step further. We also did genomic prediction. And in this case, grain yield, well, was written as grain number times grain weight. Grain weight was not, let's say there was no G by E for grain weight, so we could work with average grain weight. And if we then want to predict grain yield for future environments, we mainly needed to model the G by E in grain number. And the G by E in grain number in the end could be modeled by three environmental variables: night temperature, intercepted radiation, and soil water potential, in particular growing periods. And what did we do? Well, we estimated sensitivities with respect to those three different environmental drivers. And we had a number, but I mean, in the end, after some selection procedure, those were chosen. We have an intercept, the genetic main effect. And so this genetic main effect and those sensitivities, they were regressed on marker profiles, well, by GBLUP or multi-trait GBLUP. And in this way, we created, let's say, kind of multi-trait genomic prediction model for multiple trials and also for future trials. Okay, so some examples. Now let's move forward, because this framework that I showed you works reasonably well. But at this moment, let's say many of the models, linear mixed models that look at G by E, include all of the environmental information that is available, all of the markers that are available, and yeah, actually only one single parameter is measured or is estimated. And so actually it's a single kernel with a single parameter. So that's the variance component for G by E. The alternative that I showed you is, well, that you try to identify QTLs and then go for larger effects. Well, I mean, also, let's say, mixture forms of those two approaches are possible. One of the questions that, of course, about 10 years ago, 5 years ago, started to occur is, well, there are deep learning models. They are popular in quite a number of different disciplines of science. Can we use them also for G by E? And okay, there have been some competitions. And then, of course, the first question that arises is, can deep learning models, can they outperform LMMs, linear mixed models? And also, well, I think it's also useful maybe to try to combine the best of both worlds. So we've been looking a little bit at that, and we call it, well, our approach is called structure interaction neural networks. This is from a paper that you see over here. This is another paper also on arXiv. This paper is accepted for publication in genetics, so I guess will soon be online in genetics. The basic idea is very simple. We noticed that when you analyze a number of trials for a set of genotypes, that the main effect for the environment is always occupying about 80% of the total variation. And the genetic main effect, something between, well, 3 and 5% of the total variation, and the G by E, something between 3 and 10. Which means that the largest part of modeling phenotypic variation actually is done by environmental main effects. But this is exactly not the part that is of, well, is of less interest to genetics and breeding. So if you allow your deep learner to use end-to-end learning, which means that it tries to estimate all of those, well, in a sense, it doesn't distinguish these parameters, but if you use end-to-end learning to predict new genotypes and new environments, then most of what you will be doing is trying to capture the average difference between environments. So to preclude that, we thought it might be wiser to split up the task in 3 and first use a mixed model analysis to identify genetic main effects, environmental main effects, and G by E. And then we have 3 different deep learning tasks. So the first is to model the genetic main effects in terms of markers. The second, to model the environmental main effects in terms of environmental drivers. And then we use the representations for the genetic differences and environmental differences as a starting situation for the G by E. And in the end, we reconstruct the prediction by using, well, an estimate for the general mean, an estimate for the genetic main effects, based for the deep learner for those effects, the same for the environmental main effects, and then finally we add also the G by E part. So in deep learning terminology, this means that the loss function for the genotypes is separate from that for the environments, and it's also separate from that for the G by E. There are 3 tasks that are accomplished separately. And if you would use a standard deep learner, you would try to do all of this simultaneously, which is not a good idea, because then the deep learner will be mainly trying to understand the difference between environments and will be very happy when it does so for 80-90%. and the genetic effects and the G by E, let's say, will be underrepresented. Well, this slide is mainly to impress you, so don't try to read it. The main thing I want to say here is that we used a very simple deep learner for each of the three modes. For the genetic main effects, we simply used a multi-layer perceptron. We did exactly the same for the environmental mapping. We also used a multi-layer perceptron. We experimented with far more complicated things, let's say RNNs, CNNs, LSTMMs, etc., even language models. But in the end, these simple multi-layer perceptrons, which are, let's say, a generalization of principal components repeated a few times, do perfectly well. And we got quite good results for the genetic main effects, that's for the standard genome prediction. We also were quite able to represent the environmental main effects. And for the G by E, we took the final representations from, let's say, the genomic mapping and the environment mapping, and we created a kind of bilinear products using 64 terms extracted from the genomic mapping, another 64 from the environment mapping, and we tried to combine those as good as possible to find products of genomic and environment mapping that led to prediction of G by E. So here you can see that once again, and once again, this is not, let's say, to make you read this whole slide. This is mainly to show you that this deep learner, which is a hybrid of linear mixed model and deep learning, the end result can be represented once again in a kind of kernel mode, which is exactly the same as the mixed models that we were used to already. The ones that we were using, and I say, in the analysis of G by E trials, multi-environment trials, before we were using, deep learning. Because basically, the kernel for the genotypes is defined by the genomic encodings, that for the environments is defined by the environmic codings, and then for the G by E, we mainly combine the genomic environmic codings, let's say, to have these simple products, optimized products, to arrive at G by E. This looks extremely similar to, let's say, the model introduced by Harkin, but now with a deep learning architecture in the background and a little bit more flexibility. We apply this to a few data sets, and let's say the major data set is taken from the Genome to Fields maze competition. I have to say that it's not very easy to find data sets in the open domain, because if you want to do this, I do think you need at least 50 to 100 trials. Otherwise, it doesn't make any sense to try to use these deep learners. If you want to do anything with deep learning in relation to G by E, it will not do with only 5 or 10 trials, which actually most papers are using. So this is a large data set. Probably many of you know it already. It's about 5,000 hybrids. We have a test set of about 500 hybrids. We used over 200 training environments, and we have 26 new test environments, which is mainly a holdout year. So, and well, it's interesting that when you compare the results of classical machine learning, mixed models, and deep learning in the competition, actually the mixed models did best. This is the root mean squared error, and this is the within Pearson correlation coefficient between observed and predicted. You can see that for both, criteria of performance, actually here a mixed model and here some mixed models do better than anything that is related to machine learning and deep learning. Well, our proposal is over here in yellow, and well, it performs indeed better than anything that was there. I have to admit that yes, it did better, but it was by a slight margin. But I think the good thing is we use this for 3 or 4 big data sets already, and on all of those data sets, it always performs at least as good or better than any other competing method. So to some extent, we're confident that our proposal is not too crazy. And the other thing is, well, it's a relatively simple thing to do. Now, another nice thing is that, of course, the criticisms on deep learning are that it's uninterpretable, black box, etc. But with a little bit of post-engineering, you can get importance measure for your features. You can do this for the SNPs. You can do this for the SNP times weather interactions. You can do it for the SNP times soil interactions. So in this way, you get a reasonable feeling for what's going on. And you can also do it, let's say, for the environmental features. So as you can figure out which environmental features are important with respect to the main effects, and also which environmental features are important with respect to the interaction part. Okay, that's for the deep learning and the standard mixed models. Let's move forward now. We have been experimenting with covariates because, well, we want to include environmental drivers that explain differences over environment between genotypes. Well, the standard methods allow you to include a few or all of them, like the Harkin approach. We want to be somewhere in between. And for that, we have been looking especially at random regression. Now, this is a project together with the University of Queensland and the Grains Research and Development Council. This is about 1,000 trials in Australia, and they are all over. I can see that is in the west, south, west, southeast. So this is a large data set for which we had about 60 environmental covariates, defined as 15 environmental covariates, soil, water, but also simulated by AppSim using crop growth simulation, and then taking 4 growth stages and accumulating, we end up at about 60 environmental covariates. We made a pre-selection. We used a lasso for each of the genotypes across all of the trials. And then the environmental covariate that were most often identified with a large coefficient in the end were chosen. We then end up, okay, I have to go to the next one. We end up with a random regression model. And this random regression model, well, actually, in this case, it's one covariate, linear and curvilinear term. This is for the environmental main effects. This is kind of residual. And for the G and G by E, this is a genotypic intercept, and these are, let's say, coefficients, slopes for each genotype with respect to the linear and curvilinear components. There's a residual term. We can do this for 1, we can do this for 2. Actually, we could do this for up to 10 covariates. If you do all of that, you end up with a very large matrix that contains the variances and covariance between the genetic effects for the intercepts and slopes. And for that, we used an extra trick. We put a factor analytic structure or used rank structure on those covariates. Well, if you do that, then you have a possibility to include a substantial number of covariates in an attempt to get a decent representation of the environmental drivers for the G by E. And well, we have to say, have to go to the results. If you look at the results, I'm not going to talk you through it. There's too much. Where you see the red arrows, these are models that used 8 environmental covariates, and on average they did best. But I also have to say once again, this was by a very small margin. We have been improving on those models. It now looks slightly better. And so the models, well, are quite able to do complicated things. I would say we don't see that back too well in the final results, in the predictions. Okay, let's move to the dynamic part, because I see I only have 5 minutes left. I need a little bit more. I need 10 minutes. So I think we'll be able to get there. Okay, let's think about what's happening with respect to longitudinal information, because here I give you a simple cartoon. These are 3 situations with exactly the same final yield. Let's say that this is biomass, and let's take that equivalent to yield. But you see that I get there by very different trajectories. And so we think that if you have access to this information, you should do better with respect to GBE than we were able to do in the past. And for that, well, we have different classes of models. Now, classically, crop growth models were used that actually model the change in biomass as a function of biomass up till this moment and anything else, phenotypic traits, environmental drivers, and genotype-specific coefficients. But for use in genetics, the problem with those crop growth models is that they contain too many parameters. We cannot calibrate 600 genotype-specific parameters in breeding trials. And so we need to find a way to use the dynamics, but in simpler types of physiological models. And one of the main things I think is what are the times at which things change? So, for example, you see a bump over here. Okay, apparently something happened in the environment of which we need to take care. And it starts here and ends there. And the genotypes, well, they might act differently in this particular time, okay, and in this time trajectory. And I think that is what we're up to. So actually, we've been looking at 5 different classes of models. We've been looking at the whole genome prediction model, crop growth model, the whole genome prediction crop growth model. I'll show in a moment. We've been looking at splines, which is a very flexible regression way of dealing, let's say, with the observed data. And then mainly we try to identify the times at which things change. We try to find out what makes the changes. We also try to figure out how much the genotypes differ in their reactions. And all of that, the timings and the reactions and also the environmental drivers, they could be introduced as genotypic covariates, environmental covariates in linear mixed models, which brings us back to the framework that I already showed you. We can also go directly to dynamics because instead of using a full crop growth model, we might use simpler models. We could go from scratch and try to understand the biology from scratch. We could also use something that is not totally scratch, but still tries to identify from a multitude of environmental drivers, which are the main ones. And finally, we can use neural networks that are kept at their place by loss functions that are inspired by biological models. We will see in a moment what we mean with that. So, okay, I don't have time to walk you through all of these slides, but the whole genome prediction crop growth model, you might know because I think it's one of the most used options at this moment. Basically, what it does is it takes a crop growth model, then you insert the environmental dynamic information, you run the crop growth model, and you try to optimize marker profiles with respect to a limited number of traits. And so there are yield components, typically 4, and those are written as a function of marker profiles in a number of iterations. And the only thing dynamic here is the environmental information. Okay, I mean, I think this is a good approach, but quite costly. As an alternative, we started to look at splines, and this is work we did a number of years ago with ETH Zurich. We're still working with them. So this is a dynamic spline model. We fit a population spline. We fit deviations for each genotype. We fit plots or plant deviations from the genotype. So there's 3 levels. That's a 3-level dynamic model, spline model, actually functional dynamic model. And the important thing is that, well, we have here the individual functions for each genotype. These are the first derivatives and these are the second derivatives. And this set of 3 plots allows us to figure out what's going on. We see that where changes occur, well, apparently something happened in the environment. And when we followed that up, we could see it over here. Here you have superimposed the amount of water available. And we could see that, well, there was a dry period for this first bump. Here are once again the first derivatives. Here there are 8 different groups of genotypes. Well, they clearly have different dynamics. They have different first derivatives, and they have different amount of genetic variation for their individual genotypic slopes. So this is our building material for finding follow-up genotypic specific parameters and environmental drivers that allow us to be to use linear mixed models with this information incorporated. Now, the other option that we also tried to develop dynamic modeling using differential equation from scratch. Once again, I'm not going to talk you through the detail. The main thing here is that we assumed that the growth of biomass was a kind of logistic model, as you can see it here, but the rate coefficient, the growth constant in logistic equations, which is constant, is here a function of temperature. If you do that, you try to get a closer approximation to the observed growth patterns. And this would allow you directly to extract meaningful information from, let's say, the differential equation, because each of the parameters in the differential equation is a genotype-specific parameter that can be regressed once again on marker profiles. And with that, and you would be able to predict from marker profiles and environmental drivers to new environments. Here's one that combines biomass and water. This is the change in biomass. This is the change in water. They're coupled. You see here, let's say, the situation over time for both of them. And once again, this is an introduction to doing this for each genotype. But then in the end, you need to do this for large numbers of genotypes. So we also wanted to fit those types of equations for many genotypes across many environments. Well, for that, we developed an hierarchical Bayes model in which each of the parameters, so that's the growth rate, that's the final asymptote, the final biomass, all of those, once again, could be functions of linear functions of genotype environment, but also genotypic covariance, environmental covariance. And so these models allow you to directly extrapolate to new conditions for new environments. Here we see an example. This is for 4 environments, wheat in Australia. Well, you can see that the amount of variation changes between those 4 trials. There were about, I think, 200 trials that we looked at. The thing to realize here is that if you look at the averages for each of those 4 environments that you see here on the left, the environment is quite important. This is the environment curve, and you see that the genetic variation around the environment curve is not too large. Well, so nevertheless, if you analyze those data for G by E, it looks enormous, but when you bring it back to this kind of formulation, the G by E disappears in the differential equation. Okay, then finally, we get to the sparse identification of environmental drivers. Here, we directly model the change in, for example, the weight or the biomass and the leaf area index. So we have 2 state variables that are measured on the plants, time 1, 2, etc. So the changes from time 1 to 2, etc. Well, these changes, they are modeled in a kind of regression on an explanatory set that contains information on environmental drivers, but can also contain information on other phenotypes. But to keep it simple, let's say that we have temperature, evapotranspiration, soil, and accumulated temperature. And those are introduced in this design matrix here. And from that, we try to identify the best ones that explain the growth in especially weight. And the result looks like this. So here we have biomass over time. We have also leaf area index over time. These are the predictions for held out years. So these are 4 different years in a series of 12 for 20, potato genotypes. And if you integrate the biomass curve till the end, or you take the mean, the maximum over here, you have an estimate for biomass in that particular year for that situation, for that genotype. Here you see this for 5 genotypes for a number of years, and you see actually that final biomass can predict it really well from this approach in which you identify the environmental drivers for biomass changes directly from a set of, candidate environmental drivers. Okay, finally, because this is the penultimate slide, another way of doing it is using a deep learner, but the deep learner, can have a relatively simple structure, but we inform the deep learner that we always want a kind of logistic curve to come out. So as long as we keep the deep learner close to logistic, it's able to figure out what is happening. Actually, what it did is mainly trying to reinvent growing degree days. So it tries to figure out how many growing degree days a genotype needs. And from that, it can also predict the growth of biomass in the next year. Okay, this is the final one. So what have we done? We've been looking at linear mixed models. We have genotypic and environmental covariance. We have seen a special example with QTL by E, and we also saw an example in which we use it for genomic prediction. We looked at deep learning, in which we used a kind of hybrid between linear mixed models and deep learning, because, we split up the deep learning task in 3 tasks for genotypes, environments, and G by E. We looked at random regression and how we could do this for a number of covariates simultaneously. And finally, we looked at different ways of modeling longitudinal phenotypes And all of them, I think, are useful, but it depends a little bit on the amount of data that you have available. And I think that's it. And I probably stop sharing my screen.
Okay.
I'm not quite sure where I am now because I don't see.
Can you see me, Professor?
Let me check because I don't see anything. Yeah, I see you again.
Okay.
Yes.
Thank you so much for the brilliant presentation, Professor Fred. We'll now move on to the questions. Okay. We received some questions in the chat, but due to time constraints, our team has selected some of them for this section. Okay.
Okay.
Okay, just a second. The first question is from Bruce Paris. Are the analytical models and statistical methods for G-by interaction taught in academic curricula aligned with the tool and decision-making workflows currently used by commercial breeding programs in your opinion?
Yeah, very good question. Because, I think, okay, if we're honest, what I showed you in the last part of the presentation on these differential equations, this kind of modeling we're doing together with a number of potato companies in the Netherlands. But this is still, I think, at the initial stages. And we first have to figure out whether it works, but it looks, well, it seems to work relatively fine. But it's certainly not the case that those potato companies are using this kind of technology at this moment to make the decisions. I can say that, let's say, for the whole genome crop growth model thing, I do think that, for example, Corteva and Bayer Monsanto, they do to some extent use this technology.
Okay.
I can say more about it, but I guess that also other questions need to be answered. So.
Okay, yeah, sure. So the next question is from Bruno Trevisan. Great work. May I ask how the envirotyping was done in your LMM analysis? Were the 15 environmental covariates obtained from a public database or repository?
Yeah. Well, let's say for the Australian data, we did a few things. First, I think the Australians do have their weather records going back about 100 years for each position in Australia. So we went back to databases for all of the elementary weather information and to some extent also for soil information. From that information, which is typically on a daily basis or even more higher resolution, we more or less comprehended the information in 4 intervals that were chosen with physiologists. I mean, they determined, well, okay, from then to then is more or less the period that can be considered to be a certain physiological growth stage. So these were the ones that we used in that random regression work that I showed. But beyond that, there's a separate paper which is also on the slide, which is led by by one of the researchers from the University of Queensland, in which we did envirotyping using the same information, but then using AppSim crop growth simulations, let's say, to find, well, which trials can be considered to be part of a certain envirotype. And in that paper, you can also check to which extent that envirotyping is able to explain the G/E retrospectively. Yeah.
Okay, thank you. And another question, what model would you suggest to predict yield in missing plant occurrence to determine data quality when phenotyping across different locations?
I'm not quite sure whether I understand the question. So the question is, if there are missing, can you use masking within existing trials and genotypes to figure out whether the quality of the phenotyping information is okay? Or?
Yeah, what model would you suggest to predict yield when missing plant occurrence to determine
but
data
all
quality.
of these models actually, they are not very sensitive to missing information because in a sense, both the splines and the differential equations more or less interpolate. You don't need to have information for each genotype. You don't need to have information for each plot. All of these methods also work fine when, let's say, part of the data is missing. For the regressions, it's another story, indeed. If you want to use the linear mixed models with environmental covariates and genotypic covariates, most methods, most software packages won't allow you to have missing values in the covariates. So then, you can use different ways of first imputing the missing information. Typically, what works reasonably well is take the whole matrix of environmental covariates by environments, of environment by environment covariates, and use one of these standard imputation methods to fill in the holes in such matrices. Typically, I mean, regress one column on all the others, fill in by the predicted values, go to the next, and iterate this until you don't see any change anymore.
Okay, thank you. And the last question, with the advance in variotyping and artificial intelligence, do you believe that the way we study and model G/E interaction will change significantly over the next decade?
Interesting question. Actually, I think yes. I mean, I think we have, more advanced modeling possibilities. And because we have those, we can also design the experiments in another way so that we can optimally benefit from these improved models. I mean, if you have a very simple car that never goes beyond 100 kilometers per hour, it doesn't make sense to go to a race circuit. But at the moment that you have an advanced car, you can try out how fast you can go. So I certainly think this is the case for the integration of statistical, mathematical, and deep learning methods. I mean, we haven't spent very much, you have these things on sparse phenotyping, sparse genotyping, but I think that is just, first look at what we need to do to be able to better do the dynamical modeling. Because for the dynamical modeling, we mainly need to have the observations at the times that things are going to change. I mean, it doesn't make sense to do a lot of observations when we're at the beginning or at the end. Well, we're at, let's say, at the asymptotes. But it's not so easy to figure out when the genotypes are going to make their changes. So, but okay, I do think that a lot can be gained there by checking what the models need in terms of information to give to be able to lead to optimal decisions.
Okay. So thank you, Professor Fred. Unfortunately, our time is up. We need to move on to the next talk. Okay.
I wish you a lot of success. Thanks.
Thank you. Thank you very much to all participants for your active engagement. And once again, a huge thank to Professor Fred for sharing his expertise with us today. I hope we can have you visit us in person someday.
I'm sure you can.
Thank you.
Okay. Thanks.
So, okay,
Bye.
we kindly ask everyone to migrate to the next lecture using the link that has been shared in the chat. See you all in the next session in a few minutes.